I'd like to introduce the organizer and moderator of this webinar, Dr. Sandeepa Gautam. Dr. Gautam is a Cooperative Extension Area Citrus IPM Advisor. She is with the UC IPM statewide program operations and UC Agriculture and Natural Resources at the Lynn Cove Research and Extension Center. So welcome, Sandeepa, and you can go ahead and share your slides.
Good morning, everybody. Welcome to our webinar, which is Citrus Leprosis Threats, Mites, and Mite Management. We have about 2.5 hours of content prepared for you on this topic from various experts working in this area. Dr. Chris Godfrey at UC Davis, Tara Pittman at UC Davis, Dr. Ellen Tassi, and Daniel Carillo, also from University of Florida. Dr. Carillo couldn't be here with us today, and Dr. Ellen Tassi would be covering his presentations for us. I will be your moderator for today. We have several speakers lined up for today. I'm the first speaker. My title is pretty long here, Introduction to Citrus Leprosis Disease, Threat to California Citrus. I'll also talk a little bit about pest and predatory mites in citrus, and a little bit about mite management, the best management practices for managing mites in citrus systems. Let's begin with the most important question first. Why are we concerned about citrus leprosis disease in California? For a disease to flourish, 3 different conditions are necessary. You can see a disease triangle here. We need a host, and citrus is a host, so we have the host. We need an environment, which is weather conditions that are favorable for the disease, a vector, or something that would cause a disease transmission, which usually is a vector in vector transmitted disease. We may have mite species that are able to transmit this disease, and we surely have the environment under which the disease can flourish. What we don't have is the pathogen. So this makes it a risk for our citrus grower. It is also most devastating viral disease of citrus. It's very costly to manage. Brazil loses a lot of money managing this disease every year, and estimated crop loss in Brazil is about 20 to 100% from dieback and premature fruit dropping. It is also rapidly expanding in citrus growing regions of the world. It affects all citrus varieties, all rootstocks and scions, and it is vector transmitted. There is no cure for the disease once the disease is transmitted. The only way to manage the disease is to manage the vector. And it has multiple hosts. It has been reported in ornamentals like orchids and hibiscus. And what that means for us is there are multiple pathways of entry for this disease to California. It could come in an ornamental plant and then be planted in your front yard, and where you also have a citrus tree. And if you have a vector mite, it could move from one host to another very easily, which is why it is a risk for us. So what is this disease? It is non-systemic viral disease caused by citrus leprosis virus. Non-systemic means that the disease and its symptoms are localized at the point of transmission, usually where the vector fed for citrus leprosis disease. The viral agents cause chlorotic and necrotic lesions of citrus leaves, fruits, and twigs and branches. There are currently 12 reported viruses from 3 different genera that can cause citrus leprosis-like symptoms. For the disease itself, there are 2 different types. Cytoplasmic type, which is the most common one, and the nucleus type. Now, these 2 different types of diseases are caused by different types of virus, but they could look very similar. And like I said earlier, the virus are transmitted by Leprosis species, commonly known as flat mites, which are known to present in California in citrus system and in other cropping systems. So where is this disease now? This disease has been known to be, like native to South America, Central America. So it's present there in South and Central America. In North America, it has been reported from Mexico and also from Hawaii. It was known to present in Florida in 1968. It was first reported from Florida. But then after 1980s, this disease has not been reported from Florida, and it is believed that the harsh weather conditions, freezing weather conditions in Florida during the 1980s might have killed all vector populations, and the disease is eradicated from there. The newest report of its new distribution is from South Africa, which was reported in 2019. As for host strains, I mentioned a little bit of about it before. It goes to all types of citrus rootstocks and science. It can affect every citrus variety, every rootstock, and every sign. And other hosts, very common one, are hibiscus species, Dieffenbachia species, and orchid in orchids. And in orchid, it's known as a different with a different name. It's known as a orchid flake virus. Um, and like I said, we are concerned as California citrus growers because sweet oranges, mandarins, and grapefruit are known to be the most susceptible varieties. Now let's talk a little bit about the symptoms of citrus leprosis disease. On leaf, the symptoms are smooth and yellowish and somewhat circular in reason. And that is because the disease is not systemic, it's non-systemic. So the virus is injected via vector and it just multiplies and gets localized in a region. And wherever the virus is localized and is producing the toxin is where the disease symptoms is shown. Two different symptoms can be found. The cytoplasmic and nucleic citrus leprosis virus can cause different symptoms, but they do look similar, similar to, like untrained eyes. The cytoplasmic virus can cause lesions that appear as depressed necrotic halos or necrotic spots with chlorotic rings. So it's dead in the center, but has a yellowish halo around. That's what a cytoplasmic strain would cause. Whereas the nuclear type leads to lesions with three regions: a necrotic center, an orange colored halo, and an outer chlorotic halo. The lesions and the could also be on the stem. Wherever the virus feeds, the lesions will develop, and also on the fruit. Early symptoms on the fruit, you can see chlorotic lesions at the point of vector feeding. Those symptoms, develop individually, and as the virus, as the vector feeds more, the virus transmits from has more than one entry point, and as the lesions develop, they could coalesce and turn into this necrotic looking symptom. Severe infestation can cause a lot of fruit drop and destroy fruit drop. Secondary pathogens could affect the fruit. It could lead to branch death and tree death if the disease is not treated. How is this disease spreading? It is spread by a vector, commonly known as flat mites. There are several species of flat mites that transmit the disease, or there are several species of flat mites that have been understudied for its ability to transmit the disease. Once the virus is acquired, it stays inside the mite body, and when the vector is feeding, it gets transmitted to the plant. It doesn't transmit through the seeds. It can transmit through the graft union, but again, it's because it's not systemic, it will just stay where the graft union tissue is. The vector for the disease is a small mite known as flat mites because their body is flat, somewhat triangular, as you can see in the photo over here. All life stages, adult larvae, protonymph, deutonymph can acquire and transmit the CLS disease. It is not, however, transmitted through eggs, so it's, the mite has to feed to acquire the virus. The mite life cycle is about 3 weeks, and disease transmission, if the tree has a disease, then it can spread really fast if the mites are not controlled. And for us, prevention is easier, cheaper, better than cure. Because California's climatic conditions are highly suitable for establishment of the citrus leprosis disease, it is a risk for us because vector may have been already present in California. Dr. Chris Godfrey and Tara Pittman and Elaine Tassi and Dr. Carillo are going to cover this in detail in their presentations. For us, preventing citrus leprosis from entering into California is much easier than trying to eradicate it once detected. Or if the disease is present in some isolated areas, curing, eradicating in that isolated areas is the most effective way to manage disease from spreading into a larger area because it becomes tougher to manage then. One way to do that is avoid or regulate the transportation of propagation material. If you see anything that looks like a citrus leprosis symptoms, bring it to your regulatory authorities, to your county, to the UC Cooperative Extension offices. Okay, pest mites of citrus. There are several species of mites that are, pests of citrus. Not all of these mites are economic pests. They're more of a secondary pest, but they can be occasionally problematic pests needing a targeted treatment. Some of those species are citrus red mite, citrus flat mite, two-spotted spider mite, and citrus bud mite. So let's start with citrus red mite. It is among the most important economic mite species of citrus in the San Joaquin Valley, where most of the citrus is grown. The mites seem to flare up in response to pest management practices that tend to rely on conventional broad spectrum products. So, if you're using opies and carbamates and pyrethroids and some of the broad spectrum products that would affect natural enemies, then red mites tend to flare up. They are small mite, about 0.5 millimeter in size. They have red globular body. They have all these hairs sticking out. They move fairly slow. And because their mouth part is modified to look like a spear, like a pencil, what they do is they stab the tissue and they feed on the sap that oozes out. They slurp it out. And in doing so, they cause several stabbing and cause all this stippling that you can see on the photos over here, a silvery stippling all over the leaves and all over fruit. The threshold for these mites is pretty high, about 8 mites per leaf, and it's more like a spring pest, this species particularly, because their population cannot sustain temperatures higher than 95 degrees, so it kind of disappears in the summer season. And there are several predatory mites, virus in the summer, and acaricides that are effective against red mite. The identifying character for this mite species is that it has red globular body with the hairs sticking out, as you can see in this photo. They can be found on both upper and underside of the leaves, and they move fairly slowly compared to other mites. You might need a hand lens to see them, clearly. About 10 to 20 times hand lens would allow you to see the mites clearly. You will probably find their immatures and eggs right where there are adult mites. Eggs are reddish in color and round with the hair sticking out. And immatures are similar to adults, but just smaller in size. The other species of mites that is commonly found in citrus, again, it can be a pest in some years, especially when it's hot and dry conditions that these mites tend to like. The species is Tetranychus urticae. They're commonly known as two-spotted spider mite because of these two globular spots that they have on their body, which is actually their fecal matter pressed towards the side of the body as they eat. It's the pieces that's pressed towards the side of the body. That's what gives it the spot. So you wouldn't usually see dark spots in immature mites. It's more common in adult mites. Like with the citrus red mite, they also cause stippling on the leaves. And one of the identifying characters for them is they like this webbing. As you can see in the photo over here, they make a web, and you can usually see a lot of spots hanging on the web. And if you look close enough using a hand lens, you can see those mites moving. There are many natural enemies that feed on the two-spotted spider mites. Six-spotted thrips is one of the common ones. It's fairly very common in almond orchards, but we don't usually see it in citrus orchards. Lacewings, predatory mites, and all other natural predators would feed on two-spotted spider mites too. And acaricides are used occasionally, um, usually in spring and early summer. The identifying character for this two-spotted spider mite is that it has an oval body, like I said, with two distinct spots on the sides. They are relatively larger compared to other mites, similar in size with the red mites, larger in size compared to flat mites. They usually prefer lower side of the leaf, but they can be found on upper side when the numbers are high. And if there is one mite, there could be 20 in about a week. And as I said earlier, immatures may not have a dark spots because the food accumulates as mite develops and the spots develop with it. This species also has males, but males are more slender and relatively smaller in size. And like with the red mites, you're going to see immatures, eggs together in that same colony. Eggs are usually found on the web. They're very circular, translucent, and it's just hanging out on the web that they make. Another species, which is also the topic for today, is citrus flat mite, commonly known as Brave-Pulpus mites, and there are several species of Brave-Pulpus mites that have been reported from California. This study is from 2013, published in Citrograph, showing percentage of flat mites in citrus collections. The most common species was Brave-Pulpus californicus, followed by Brave-Pulpus lewisi. In the Californicus was common in coastal region, in Imperial Riverside region, whereas in the valley, Brave-Pulpus lewisi was a more common species. This is more of a secondary invader. It likes to feed on scar tissue already, either a mechanical scar or a scar that's made by another insect, and it just likes to feed and hide under there and develop in there. And although it's not a feeding symptoms, but it causes scabbing because they like to suck around that area and make that symptom look like a scab. There hasn't a treatment threshold that has been established, as it is not a primary or economic pest, it's a secondary pest. But it can be an export concern because it is a potential vector. Some species of Brave-Pulpus mites are potential vector for this citrus leprosis disease. And occasional acaricides may be needed, but whatever is being used in citrus systems usually ends up controlling citrus flat mites, and we don't usually see a need for a separate targeted treatment. Identifying characters, if you do see these flat mites in your orchards, they are very small. They are distinctly flat. Unlike citrus red mites that have a globular plump body, they are very flat. They have somewhat triangular body, and they like to be present on depressions, like where there is a depression on citrus fruit, that's where they like to be. Or like I said earlier, they like to make their colonies around a scar tissue, and it stands out because their color is somewhat oranges yellow in color, and their eggs are similar in color, so when there are a lot, it does stand out. Another species that has been reported in California is citrus bud mite. It's a different family, Iridophydae. They are more elongated mite. They, as you can see in the photo here, they're more like how do I say this? Have an elongated body like a comma, like that you see in your keyboard. They're very small in size as well. Their body size is about less than a quarter of a millimeter. Um, they're tapered around the posterior end, but all four pairs of legs are around the mouth part. And these mites can cause fruit distortion because they feed on the developing bud when that fruit is setting, and their feeding can cause to distortion of lemon fruit, as you can see in the photos over here. And these mites are more problematic in the coastal region. Have not been It's not a common species that we usually see in San Joaquin Valley. And also, for this mites, threshold has not been established. Levels as high as 80% bird infestation have failed to cause consistent or predictable economic losses. So they can just be there, not do any damages, and they're very tough to monitor because they are tucked in within that growing bird. So usually you would see a symptom on the fruit, and backtrace it to the mite. But like I said, even 80% of bird infestation have failed to cause any consistent or predictable economic loss. Other pest mites that are reported from California but are not prevalent in the San Joaquin Valley are broad mites, um, Kaunza spider mites, and Tetranicus six-spotted mite, as you can see in this photo, and Texas red mite. Okay, now let's move on to the management. I'll be very quick in talking about management. Water stress trees that are, especially in hot and dry conditions, are more prone to mite infestation. So irrigating your trees properly will help with, any mite infestation. Even if infestation is there, trees can recover. Dust covered trees are very attracted to mites, and they can flare up, so water the roads to settle dust, drive slowly in the orchards. For physical, pressure wash can help, especially with small trees. Biological, there are predatory mites present. There are six-spotted thrips. There are generalistic predators like lacewing, big-eyed bug, minor pirate bug, six-spotted thrips, and whatnot. For chemical, acaricides do a pretty good job of controlling mites. Like I said, predatory mites pretty commonly found in citrus systems in California. One of the most common species we see in California citrus systems is UC tularensis. It has a tear-shaped body, and it can feed on different species of mites and also on thrips. And depending on what it is feeding on, their color can be different. They can look reddish in color if they're feeding on red mites, and they can look very translucent in color when they're feeding on citrus thrips. The other species are Prunimatus ubiquitous, Phytocis persimilis, Tiflodermus occidentalis. These mites are also present in California citrus systems, and when they are present, they will attend to the pest mites. Acaricides, I'm not going to go over a lot of detail. What you can see over here is Fujimite, Nielta, Magister, Nexter, Invitor. All these acaricides are effective in controlling citrus red mites. Similar results just with 1% oil only. Venerate and Vito also effectively produce citrus mite populations. For flat mites, we haven't been able to do any field trial because it's really tough finding a field infested with flat mites in a consistent way to do a field trial. But we have tested acaricides in laboratory, and in laboratory, all registered miticides perform very well against flat mites. So if you're using it for controlling other species of mites and flat mites are there, flat mites are probably controlled as well. Oh, I had that slide twice. I just talked about it. So to summarize my presentation, as a host, host and the environment is conducive in California, citrus leprosis is risk to California citrus growers. If you do see any signs and symptoms and you find mites around the, necrotic symptoms that look circular and looks like a leprosis disease, if you suspect it to be leprosis, bring your samples to regulatory authorities. You can bring it to county offices or UC Cooperative Extension offices. Send me an email, call, bring a sample, drop it off. We will check and we'll get back to you. Mites are common in citrus. Pest mites, there are several species, but rarely need targeted treatments. Predatory mites are present in California citrus systems, and acaricides are effective in managing pest mite populations if necessary. That's all I have to say. If you have any questions for me, please type in the chat. I think went a little more than 20 minutes at a time for me. So I'm going to call up another speaker for today here. Um, Dr. Chris Godfrey, she's a project scientist at UC Davis, and she will be talking to us about citrus leprosis disease, identification of the disease, symptoms, vectors, and as these issues relate to California citrus. Chris, I'm going to stop sharing so that you can start, okay?
Yeah, I'm going to talk about citrus leprosis disease, and my comments are going to be more oriented at how it relates to California. And as Sandeepa said, this disease is a serious economic disease of citrus in places where it has established. Now, as she said, it's a viral disease. She said as many as 12 viruses. There's a little bit of debate, obviously. I'm going to talk about 7 viruses. And the reason for this is to identify a virus, you sequence the genetic material in it, then you use a very sophisticated statistical algorithms to tell you if it's the same virus or if they're different enough, you have a different virus. So I'm going to talk about 7. But what all they have in common is they're all transmitted by a species of Brevipelpus mites. So these viruses get referred to as Brevipelpus transmitted viruses. And okay, it doesn't want to do that. There, oops, sorry. And as Sandeepa said, they don't move systemically. Because usually if you think of a virus disease, you think of something like citrus tristasia virus, that once the aphid puts it in the plant, it moves throughout the plant. These don't do that. So if there's a mite that actually has a BTV in it, and it starts feeding, and that's called a viruliferous mite, that is where you're going to start to see the lesion. And it won't be much, it'll go a few cells beyond that, but it's not going to go throughout the plant. And once the mite acquires the correct BTV, because there's specificity between the mite species and the viruses. So not every Brevipelpus mite will pick up every Brevipelpus transmitted virus, okay? Keep that in mind as well. Once they acquire it and it's the correct virus, it stays with them throughout their life. And they are referred to as Brevipelpus or viruliferous mites, sorry. The egg stage obviously can acquire the disease because the egg stage does not feed. Now, this is a disease you can't just go out in the field, look at symptoms and go, oh yeah, there it is. You actually need plant material. You have to use molecular biology and you identify the virus that's in that plant tissue. Okay, as Sandeepa told you, citrus leprosis disease has been around a while, and it was actually first described out of Florida in the early 1900s, but the growers said they had seen it since the 1860s, or at least what they thought was the same disease. And back in that day, it was called either nailhead rust on the fruit, because those little round lesions, that's what they look like, or scaly bark because of the lesions on the twigs. And then there were years in Florida where these growers were getting 50 to 75% fruit loss, and other years only 0 to 25%. And some of this had to do with the density of viruliferous mites or whether they were using sulfur or some other method of controlling mites. By the early 1960s, you could not find citrus leprosis in the field in Florida. People debate what it is, but it was most likely a combination of mite control along with some really adverse weather conditions that occurred during that time. Now, in South America, where we tend to think of that's where it came from, well, it may have, but whatever. It was reported in the 1920s in Paraguay, and they called it lepra explosiva. That later, it was found in Argentina and Uruguay, and in 1931, it was found in Brazil. And because there wasn't a lot of communication among the researchers, it took them until 1932 to realize what they were seeing in South America was the same as what was in Florida, and they all agreed to call it citrus leprosis disease. Today, it's been reported from mainly the Western Hemisphere, except for South Africa, which was just, as Sandeepa said, found in 2019. This map from 2003 from the European Pest Protection Organization shows where the disease is located. Now, remember I told you there's as many as 7 or more viruses. That doesn't mean that all 7 viruses are in every spot on this map. In some cases, there will be multiple viruses causing it, and in others, it's a single virus. So you can see it's pretty widespread in the Western Hemisphere. What's missing from this map is actually in Florida, because 2 of the viruses that could cause citrus leprosis disease, they're found in other ornamentals in Florida, but they're not found in citrus. We know that those viruses can cause disease because of laboratory experiments. So since these viruses are all told apart, looking at their sequences of the genetic material within them, I'm going to start with the first 3. They belong to the family Kittiviridae. And the Kittiviridae are characterized by what they call positive sense single-stranded RNA viruses. And they tend to live in the cytoplasm, or the viruses are found in the cytoplasm. Now, the positive sense, what that means is these pieces of RNA that make up the viruses, they can act as messenger RNA in that plant cell to make the proteins that cause the pathogenicity from the virus. Included in this group are citrus leprosis virus cytoplasmic, which is in the top photo, and that picture was taken in El Salvador. Then citrus leprosis virus cytoplasmic type 2. And it's type 2 is a variant of citrus leprosis cytoplasmic, but statistically it's different enough it needs its own name. And then in the bottom photo, that is hibiscus green spot virus 2 on mandarin. It was taken in Hawaii. So where are these located? Well, citrus leprosis virus cytoplasmic, or CILVC, it's found commonly in South America, from Brazil all the way up through Central America and into southern Mexico. Not in Sonora, but it's creeping northward. The type 2 CILVC, it's found in citrus in Colombia, and it's found in Florida, but in ornamentals, not citrus. The hibiscus green spot virus in Hawaii is found in ornamentals, as well as in citrus. Now, the other 4 viruses that I'm going to talk to belong to the family Rhabdoviridae, and they're negative sense single-stranded RNA viruses, and they tend to be found inside the nucleus of the plant cell. They're called negative sense because you can't use that piece of RNA as messenger RNA to make a viral protein. It has to be transcribed into a messenger RNA before it can make the protein that causes the pathogenicity. And the 4 are orchid fleck dicorovirus, which I tend to call orchid fleck virus. Sorry, it's not quite right, but it's in the top photo. You can kind of see the lesions, and those are orchid leaves that it's on. This virus has a broad host range, so it's found in a lot of different plants. Then they've also found citrus bright spot virus, citrus chlorotic spot virus, and then in the bottom photo is citrus leprosis virus nuclear. So you can see the symptoms look very similar. So where are these found? Well, orchid fleck is common because it hits ornamentals as well as citrus. For citrus, it causes leprosis in South and Central America, in Hawaii, and in South Africa. In Florida, it's in ornamentals. Now, you may see reports that orchid fleck virus has been found in California. Really, it hasn't been. It's not out in the environment in California. It's found in nurseries, and CDFA goes in and does regulatory action and destroys it. So yes, it has come in on nursery stock. So there is one way it is coming in and moving. Citrus bright spot and chlorotic spot viruses are found in very limited areas in Brazil. And the citrus leprosis virus nuclear, it's in limited regions of Brazil, limited regions of Panama. And they think that this is probably the virus that was in Florida back in the late 1800s, early 1900s. And then it's been found in the past in Mexico. So in terms of symptoms, your severity is going to be dependent on your citrus cultivar. Also, what's your plant development stage? A young tree is going to suffer more than a mature tree. Also, it's the density of mites on the tree. And these would be viruliferous mites, not just mite density. And sweet orange is the most susceptible to citrus leprosis. And as Sandeepa said, sour orange, rough lemon, mandarin, and grapefruit, and some of the limes, not all, are susceptible. Lemons, it depends on what virus is going in. For some of the BTVs, lemons have some resistance. For others, no. In terms of symptoms, okay, on the leaves, in the top arrow, those are beginning lesions from the mite feeding. They're yellow to light green, they're circular. Then as lesions get older or coalesce, you get necrotic spots in the middle, which is the lower arrow in the photograph. And these are places where the mites have fed. If you get a lot of lesions, you can get severe defoliation of your tree just from these lesions on the leaves. The stem is a lot harder to see because they're tiny. And in the photograph to the left, the top, the two arrows to the left in that picture are trying to show the shallow, small chlorotic lesions. And they're fairly early on. The lower arrow, that lesion is easier to see because it's turning brownish. But they're very difficult to see on these little stems. Now, the older lesions on a stem start to coalesce. That's the arrow on the other side, the lowest arrow. And again, in the photo, it's a little difficult to see, but that's what they'll look like as they grow together. Now, when you go in the photograph to the right, you get large lesions forming on branches and trunks, and these are lesions that coalesce. The top arrow is trying to show a branch, the lower is trying to show the trunk, but especially the lesions on the trunk, they can be confused with a disease we already have in California. It's a viral disease, but it's called cirrhosis. So if you see these crusty lesions, as Sandeepa said, turn in a sample because it takes molecular biology to tell which one of these viruses you actually have. But with the cirrhosis, you're going to see it mainly on the trunk. You're not going to see it on leaves and twigs. But just know we have something out there here in California. Now, in terms of the fruit, the inside of the fruit is fine. This is not like HLB where you can't eat the fruit. You could still eat the fruit if it was your dooryard tree. But the rind has been destroyed. And from a fresh market, it's a problem. And again, the lesions start as light yellow to light green. As they mature or grow, they get brown necrotic spots. If you get any wounding of these, you're going to get secondary pathogens going into that fruit and destroying it. Also, the branch it's on could wilt from some of this feeding. You get a color change earlier in these infected fruit, and you can get fruit drop well before the uninfected fruit would drop from the thing. So there's some real problems with the fruit and these symptoms. Okay, in terms of damage, okay, the impact of the disease on your orchard is going to be related to how much leprosis you actually have in the orchard. And for a tree, that's dependent on the density of the viruliferous mite population. How many of those mites with the right BTV do you have on that tree? You're going to lose fruit, marketability of fruit because of the lesions. You're also going to lose it from premature fruit drop. If you start losing branches or vigor in the tree, subsequent years, you're not going to have the fruit production. And young trees can actually die from too many lesions and too much mite feeding on the trees. In areas where leprosis is common, they have millions of dollars worth of damage. And most of that damage is on sweet orange and mandarin. And in Brazilian citrus, this is the entire citrus industry, but controlling the mites, which is how you control this disease, is estimated to cost them $80 to $100 million every year. So this is a substantial economic loss. The spread is mainly by moving viruliferous mites on any kind of plant material, whether that's a box of fruit, a nursery tree, anything like that. And you can accidentally plant infected plant material. The grafting, since it doesn't go systemic, the leprosis is going to stay very near that graft union. It's not going to go that far. So obviously, there's a lot of regulations around the movement of fruit and plant material in areas where this is from, because we don't want to move viruliferous mites. And this can cause a loss of market, either domestic or foreign, besides an increase in cost and trying to meet all the regulations and the quarantines. For California, this could be a major impact because in 2024, California's citrus production, which accounted for 85% of all the citrus production in the United States, was valued at over $2.5 billion. 75% of all this production is exported. And our main trading partners are Canada, South Korea, Japan, and China. Canada is not going to want mites because they can infect nursery plants and things like that. So we've got to not have mites with or without leprosis. South Korea, Japan, and China are going to be more particular because they have citrus industries they're trying to protect. And if you remember the map, most of this disease is in the Western Hemisphere. So every country or trading partner, they're the ones who get to decide what plant pests are not allowed in their country. They're the people we negotiate with to figure out the phytosanitary agreements. And the receiving country, they'll inspect incoming shipments. And in some cases, if they feel the pest is bad enough, they'll come to U.S. production areas at certain times during the season to make sure the phytosanitary agreements are being adhered to. A receiving country has the absolute right to stop shipment, treat a shipment, or send it back for any perceived violation of these agreements. So there could be a real impact on California trade. So for us in California, it's not just losing yield because of unmarketable fruit or tree vigor. If we get leprosis, you're going to see increased production costs. You have to control the mites or whatever other things the phytosanitary agreements say you have to do. And initially, and we've seen this with other pests, you can have a loss of a domestic market because you won't be able to move your fruit across a county line or across a state line until all of the agreements get put into place. The export markets may also close. So it's something we don't want here. And all of these things are going to impact how you do your production. They're going to hamper the movement of your plant material, and they may even hamper the movement of equipment. Because for some pests, you have to wash out the picking basket to make sure that you're not moving the pest. So there's some real implications here. Now, the viruses that are the most concern to California citrus would be CILVC. This is the cytoplasmic virus, and that's because it's the most common virus out there causing citrus leprosis from Brazil through South America, Central America, and in southern Mexico. And in southern Mexico, it's slowly moving forward, but it's still in southern Mexico. We are concerned about cytoplasmic type 2, not so much of because it's in citrus in Colombia, but because it's in ornamentals in Florida. So some of that could accidentally move. Same thing for the hibiscus green spot virus that's in Hawaii, both in citrus and in ornamentals. And then, of course, orchid fleck virus, because it's found pretty much in a lot of places. It has a lot of host plants. And again, for Florida, it's in ornamentals. We just don't want to find it here. Now, for each of these viruses, they're pretty specific about what Brevipelpus mite species can be their vectors. So for us, the ones we're concerned about, CILVC is vectored by a mite called Brevipelpus yothersi. Cytoplasmic type 2 is also by yothersi, but also by Brevipelpus phoenis sensu latu. The hibiscus green spot virus is vectored by Brevipelpus azores. And the orchid fleck is vectored by a mite called Brevipelpus californicus. Now, I want you to know the taxonomy of these mites is not very clear, and as you can imagine, because they're so tiny. So we need to say, well, who do we have here in California? Well, the two we have in California that most people think of is something called Brevipelpus californicus and something called Brevipelpus lewisi. A further talk is going to talk about this taxonomy as to whether these are actually the correct names. But the million-dollar question here is, do the two species of mites, Californicus and lewisi, do they vector any of the BTVs that are of concern? Well, stay tuned. Tara Pittman has done research on this question, and she's going to provide some answers to this in a talk that's coming up. So thank you.
Ellen would be talking to us about Brevipelpus mite and mite-transmitted diseases in citrus and other crops. She's a postdoctoral research associate at the University of Florida.
So this presentation was supposed to be given by Dr. Carrillo, but he had a personal matter, so I'm be covering for him this time. His talk was supposed to be about the Brevipelpus mites and what are the diseases they transmit, not only for citrus, but other crops. I'm going to repeat some of the information that you guys already received. But my idea here is mostly show you the diversity and how important is this pathosystem. And with this, we can have an idea of what we have to mitigate. Okay, before diving into the problem of citrus leprosis, it is important to know that the whole of the mites as plant virus vectors. Not only Brevipelpus are vectors of viruses, there's other groups that are vectors of viruses. For example, we have the Areophidae, like Sandeepa talked about, some of them. They're also really important vectors of virus. We have a review published a couple of years ago that verse about most of this virus, if someone is interested. But the ones related to citrus leprosis are the viruses that are in the group transmitted by the tenuipalpid mites. The tenuipalpid mites are the family where Brevipelpus is located in. Brevipelpus is a genus with around 300 species today. There is some research going on, but it's in waves. Sometimes there's more, sometimes there's less. Some years there's an increase of information due to extraordinarily because of the virus they transmitted. There is a publication from 2003 that they examined using the pest taxonomy of the group, only 3 species of the mite. That will be the older Phoenicis, the Obovatus, and the Californicus, and only with these 3 species, they reported more than almost 1,000 plant species as hosts, as suitable hosts for these mites. This versus more than 140 families of plants. These mites, the flat mites that are the main center of this topic, they are especially abundant in the tropical and subtropical regions, which are also the most impacted areas where the crops are concentrated. The crops that are impacted by these mites are concentrated. What makes it important is exactly the fact that they transmitted pathogens. We have some species that are related to virus transmission, not only to citrus, but other plants. The 2 presumably main vectors of citrus leprosis is Californicus and Yodersai. We have other species here that they are also vector other diseases, some from citrus, some for ornamentals. And we have, for example, here in the list, Lewisai, that is a species that is important for quarantine purposes. Now, going back to the biology of the Brevipalpus mites, the life cycle of these mites is relatively long when compared to other plant-feeding mites. The females is going to lay eggs. The eggs normally are preferable laid in crevices, fruits, stem, lesions, or like dirty areas of the plant. They want protection for the eggs. This egg is going to take something around 10 days to hatch. Then we're going to have the first stage, that is the larvae, that is 3-legged, followed by the protonymph, the deutonymph, and finally the adult. Between these stages, we have the chrysalid stages that are immobile stages. All this cycle is going to take around 30 days, and the female can live other 60 days, in between 30 and 60 days, depending on the weather conditions. And it is going to lay 1 to 2 eggs per day in ideal conditions. The interesting thing here is that the females, they reproduce these females that are related to the virus transmission. We have to remember that Brevipalpus is a larger genus and we have exceptions. But these females that transmit, the Brevipalpus females that transmit the virus, they reproduce it by asexual teletoky. It's a parthenogenesis way of transmission, of reproduction. So if you have a single female or a immature stage following a plant, the single mite can establish a whole colony because it's going to lay the eggs. It has a bacteria that causes a feminilization, so the female is going to lay the eggs without the need of males. And they're going to keep reproducing like this without the need of exchange of gametes with males. So we have 2 main groups of viruses that transmitted in 2 families. That is the family Kitaviridae. The Kitaviridae has the genus Cilevirus and Igrevirus, with the main virus, the main known virus being the citrus leprosis virus C. This is the one that is called cytoplasmatic virus, because when we studied using electron microscope techniques, we see the sites of multiplication of the virus linked to the cytoplasmic of the cell. When we go to the other family, the other group of viruses that these mites transmitted, the Habidoviridae, we have a genus that is Dicoravirus that concentrated the virus that these mites transmitted. The type species of this genus is the orchid fleck virus, the virus that goes to orchids and also can affect citrus. In this case, it's commonly known as the nuclear type of virus. Why? Because the sites of multiplications, you can see the inclusions in the nuclear of the cell. And after the nuclear, they migrate to the other areas. When we look at a broad symptoms of this virus, not only in citrus, but other plants, what we can see is that they are really characteristic, whether Cilevirus or Dicoravirus, doesn't matter if the cytoplasmic or the nuclear one. All of them are going to produce localized symptoms, as you can see all around here. These infections do not become systemic. There are reports in the literatures of plants with systemic infection, but only when was test plants in really controlled conditions, high temperature, but like in the field, it doesn't happen up to now, as far as we know. We just have the localized symptoms. It is restricted to the epidermal and parenchyma of the cells of the lesions. They rarely reach the vascular tissues, the bundles, but from them they don't move. So they are most located in the areas where they might feed, as Chris already talked about. You can see you can have chlorotic spot, necrotic spot, green spots is a variety. And these lesions can be observed not only on leaves, but also in fruits, in some cases on flowers and on twigs and branches. In the citrus belt, in São Paulo state, Brazil, and Minas Gerais, that are the area of the country, the southern area that produces most of the citrus, the citrus leprosis is one of the most important diseases, being the most important viral disease. The control relies mainly in chemical application to keep the population of the vector low. So it corresponded to 10 to 15% of the phytosanitary costs. The problem with this as well is that we have to control the mite, and we also have greening, and there is a competition because due to the cultural practices and everything to control greening, there was changes in the control of the citrus leprosis. They start like addensating the plantations, the groves. So we have an increase of this virus in the past few years in Brazil, which increased, like what we show here in this graphic is the fruit drop, the losses of fruit due to this disease. So it's an ongoing problem, and when established, it's really costly to keep it low. When we think about distribution, when we look in all the viruses that are transmitted by Brevipalpus mites, what you can see, as I already said in this talk, is that most of them are concentrated here in the Americas. We have a variety of different viruses in different host plants. The most important one, that's the citrus leprosis virus C, is from Argentina through Mexico. Then we have C2, that is Colombia and here in South Florida, but we also have reports in ornamentals in Hawaii. And recently, there's a report of this virus in ornamental in Australia. And the only one that up to now is the worldwide of the spread one, we can say like that, is the orchid fleck virus, because it moves with the orchids that are the ornamental, that has high value. People movement these plants because of their beauty status, and with this they can end up moving the virus and the mites as well. Now going up to the virus that are transmitted by these mites, this virus, the previous talker versed a lot about this, but just to let you guys know, in Brazil we had a study, and what we could see as diversity of this virus is that we have two main strains of this virus circulating there, and one of the strain apparently is being the predominant one, like is substituting the old one that we call São José do Rio Preto, is the name of the city where it was first described. And we have the other one that is the Cordeirópolis, also related to the place where the first genome was characterized. This other one, this Cordeirópolis, is the one that occurs in all Americas, and this São José do Rio Preto is the one that is concentrated in the state of São Paulo. Then the citrus belt that has also in Minas. The important thing here is like apparently one is prevalent over the other, but we exactly don't know why this is happening. This is something that is under study by Juliana Stua and her group in Brazil. And moving on to other viruses that these mites can transmit, just going briefly over them. Most of them are associated to ornamental plants. When we think about like fruit crops and something like that, we have the passion fruit green spot virus, that is the Silvevirus passiflori, that affected passion fruit. In Brazil, this virus also can be found in several ornamental plants like hibiscus. Apparently, all these viruses like hibiscus. We also have Solanum violifolium ring spot virus, is a virus that affected some ornamental Solanaceae. And two viruses that were first described in they are described in ligustro plants, like privet plants, that they are not only found in these plants, but are also in orchids and other ornamentals. So just for you to have an idea of the diversity. The virus that goes to passion fruit, the passion fruit green spot virus, at least in Brazil, we can find it in several other ornamental plants occurring naturally, not by transmission assays. These are commonly in the landscape. One characteristic of these viruses is that normally they're going to produce like green spots in yellow leaves. So people can mistake it to just normal process of senescence of the leaves, because normally when the leaves in this stage, the mite doesn't find this leaf palatable anymore. It's not tasty for the mite anymore, so they move away. But when we apply molecular techniques, we can see that this is the virus that are transmitted by Brevipalpus mites. Another important one is the hibiscus green spot virus 2. That is the one described in hibiscus in Hawaii, that has her scientific name now, Hygravirus waimalo. It is not present only in hibiscus, but it can affect Citrus volkameriana. And one interesting thing that the group from Brazil recently found, the group of Juliana Stua, is that there we have a ancient pest disease that never had like the etiological agent fully described, was recently restudied. And what we used to call chlorosis zoonate chlorosis in Brazil is in reality the same viruses. So this, apart from being named hibiscus green spot virus, is a virus that goes to citrus. And this is lemons, limes, tangerines, and others. In Brazil, we have these present in the northern states, more hot areas. Not on the area that we have like the commercial groves, but it causes like these spots on the fruits, for example. But it's a virus to keep a look at, because the symptomatology on citrus is not that clearly necrotic center with yellow aloe that we see in the normal citrus leprosis C virus. Now moving to the other family of viruses, the genus Dicoravirus. One important one for us in Brazil, it's restricted to Brazil, this one, is the coffee ring spot virus. The coffee ring spot virus, it affects coffee, as the name says. It causes the lesions on the leaves. This is estimated to give like around 10% leaf drop. And with the leaf drop, we had the losses of photosynthesis that can reduce plant production. Another problem is that they can feed on the fruits, as you can see these spots here. And this can cause, when the fruits develops, can cause cracks. That is an entrance for fungi, and this can depreciate the value of the beverage, the quality of the beverage. There's a recently publication saying that the succulent plants can also be a suitable host for these viruses. So it's another plant that is moved a lot due to ornamental purposes. So it's another plant that we have to take a look and take care, because it's not a plant that we're going to imagine to have brevipalpus on, but apparently they like it, and they also can be a source of inoculum for this virus for multiplication. Moving on, we have another virus that is commonly described in ornamental plants. That is the Clerodendron chlorotic spot virus. These viruses were primarily found in Clerodendron hibiscus in Brazil, the first description, but it can also go to orchids, to Dieffenbachia and other ornamental plants. As you can see here, there's also possible to see some necrotic spots on the flowers. So this virus has the ability to multiply also in the flowers. So moving on, we have the citrus leprosis virus in, that is the nuclear leprosis that we described the first genome of this virus in Brazil. This is located in the state of São Paulo mainly, where it was first reported. In areas that are not like big plantations, big groves. It's like small vicinities where people have 1 or 2 trees only. And it's more mild climate, not too hot. It causes this kind of lesions, and it is transmitted by Brevipalpus phaenensis. And in a couple of years, at least the trees that were following in the study did suffer like a complete dieback of the tree. We lost the trees. So this disease, we think that is really aggressive. It's one of the reasons why it's not that established and really found everywhere around. There is other 2 decoraviruses that are responsible for citrus leprosis. That will be the citrus chlorotic spot virus, that is the Decoravirus citrus, and citrus bright spot virus, that is the Decoravirus australis. The citrus is reported in the northern region of Brazil, that is more hot climate. It has as characteristic this kind of lesions that mainly it's difficult to see necrotic spots in this kind of lesions. They are more like these big yellowish blotches in the leaves. This virus, we also see it and report it in hibiscus plants and agave plants. So it's another 2 plants to keep an eye on. And the citrus bright spot that has like these bright yellow lesions on the leaves, we found it for the first time in the south region of Brazil. It's also areas there is no commercial production, and it's more cold, mild climate. This virus is also associated, the citrus bright spot, with the transmission by Brevipalpus auris, like the hibiscus green spot virus that occur in Hawaii. And then we go to the orchid flake virus, that is the Decoravirus orchidaceae. They affect like several genus of orchids, as you can see here, causing different symptomatology. It also can affect, there is a strain, orchid flake virus C, that goes to citrus plants, different types of citrus, different rootstocks. So it's it can be widespread. It also can cause the characteristic lesions on the fruits. And we have it occurring in Florida in some ornamental plants. This is another slide for you to take a look on the variety of the symptoms that this virus can present itself in orchids. Like can be like yellow spots, can be like clear ring spots. In other cases are like more literally lesions. Without a molecular tool assay to test this, is really difficult to separate from other viruses that also occur in orchids. So this is why it is important. Here in Florida, those are some of pictures of ornamental plants that were found in the landscape infected with orchid flake virus. We have like Liriope, Ophiopogon, Aspidistra. We have pandanus grass. They are kind of these big leaf plants. And one characteristic is like the lesions of the leaves are more, they are not that clear pattern of yellow aloe with necrotic center. Here is one just for you to compare with what happens in the orchid flake virus in citrus, like how different is the kind of symptomatology. So it's something that we have to be really careful because it's difficult to spot how these viruses are moving. So association with mite and some lesions probably is worth the investigation. So what causes what we call leprosis syndrome? We have all these viruses that we talked about that were associated with diseases that can cause what we call citrus leprosis. The most important one are the three first ones. That is the citrus leprosis C, citrus leprosis C2, and the orchid flake virus is the one that are reported occurring in commercial areas. The C2 and the orchid flakes are the ones that are more close to us, is the ones that are here in Florida, the ones that we are more taking more careful of, more surveillance. The surveillance is more up on these two. The other ones also occur, as I mentioned you, but they are more located in small areas. They're not a big infection fossil of these being reported anywhere. When we look on the distribution, the black icons, the black mites, are where the mites are reported. And the red ones is where we have the mites and the virus circulating. So this shows us that the mites were present in the landscape. Sometimes not the right mite, but the mites that have potential of transmitting virus. So if we accidentally introduce it to new areas, mites or plants that have the virus that will be suitable for being transmitted with the mites that are circulating in the area, we can have a quick spread of the virus in regions where it's not occurring. Here, just for you to have an idea, this is a phylogenetic tree of celeviruses. There are some of the viruses reported in this tree. And here are the species of Brevipalpus that are associated with the transmission of these viruses. Brevipalpus yodersi right now is recognized as probably the most important vector, is the vector that transmitted the most economic important disease, that is citrus leprosis virus C. And is also related to transmitted other viruses, other Brevipalpus transmitted viruses. So it's one species that we have to be really taking care of. And another characteristic of Brevipalpus yodersi is that this is a polyphagous species, so it can feed in more than one host plant. It is widespread. Then we have the passion fruit virus, that we have papayensis as the main vector, but it can also be transmitted by yodersi. We have the virus that affected ligustrum. We have like Tucuman, papayensis being transmitted by them. And we have the Solano violefolia ring spot virus, that is another virus that goes to ornamental, that are transmitted by other species like Cobovatus, papayensis, and Ferraguti. This shows us that it's not only one species, we have like different combinations, and we have to be working or taking care of the right combinations of virus and vectors when we want to study transmission and understand what is happening. This is for the dicoraviruses. For the dicoravirus, right now we have what we can call like 3 subgroups that have 3 different groups of vectors. The first subgroup that has the orchid flake virus, we have Brevipalpus californicus associated. We know now that Brevipalpus californicus in reality is a complex of species. So we are still working on to try to determine which one is the one that is the real vector of the viruses, so we can do transmission assays and better understand what we have. The citrus leprosis virus nuclear one is transmitted by Phoenicis. Then we have Azoris transmitted citrus bright spot. The cough spot is transmitted by Papayensis and also Yodersi. The citrus chlorotic spotted by Yodersi. And the clerodendron by Yodersi. As you can see, Yodersi is kind of the best, let's put like this, the best vectors because it can transmit it by more than one disease. So as take-home message, some things to remember. Remember that citrus leprosis existed in Florida in the 1800s and 900s. It could cause between 50 to 75% of fruit drop and fruit loss. The virus are present here, but not on citrus right now. We also have mite populations. The other side is really widespread around here. We don't know exactly about the virus that used to vector this disease in the 80s and 90s. Is something that we're trying to using herborized material and slight collections of mites, try to trace back what is happening so we can compare with our actual situation to do some risk assessments. We have three main viruses circulating here. There are the orchid fleck virus in the ornamentals that I talked to you about. There is the Riope, Smilax, the Pandanus, the Aspidistra. We have two orchid fleck strains in orchids circulating. As Chris told you about, some orchids, genus of orchids, were reported with this virus, although it was in controlled conditions and greenhouses. It is present, so it shows that it can move around, so we have to be really attentive, really take care of this. And we have the hibiscus infecting Cile virus, that is citrus leprosis virus C2 in hibiscus. It is really widespread in all the state of Florida. It's another way of this virus moving. We have some transmission assays to try to understand if this virus can go to citrus. It's an ongoing study. Just for you to have an idea of how do we take care of this disease in Brazil, how is the monitoring and everything when the disease is already established. What do we do? We do a systematic monitoring strategy to detect citrus leprosis early. That's the idea. So we do a sampling of 5% of the plants and select 1 out of every 10 trees in alternate rows. From each plant, we take 3 to 5 fruits or branches to inspect it for symptoms every 7 to 14 days. Because the regular monitoring helps the initial detection and guide timely for management. Because in 30 days, this disease can be showing symptoms. And once this one mite is infected, it can, imagine every time that the mite pierces for feeding, it's going to do a point of inoculum. So the amount of damage that a handful amount of mites can do is really harsh. Another important thing in Brazil is understand which part of the plants should be monitored. Previous studies, we saw that the mites were concentrated mainly on the top of the canopy. So this is where most of the sampling is located. If there is no fruits, we try to inspect at branches. And the action threshold is 1 to 5% of the fruits or branches with mite, the presence of mite. The chemical control is purely, the control in reality is pure acaricides. We have some acaricides that are registered. I'm not giving any suggestion, any recommendation. But what we try to do there is using these acaricides like in different groups, in different applications to avoid resistance, because it's something that also happens in Brazil, that mites creating resistance to the products. And we also have some cultural practices that are important. That is reducing dust, promoting no-host cover crops, like we have to avoid using cover crops that can be host of these viruses, otherwise we have a inoculum source inside our plantation. Also, controlling scab and scales is important because this is what mites like to put their eggs in because it protects the eggs. It's also important to disinfect and harvesting material. We do sanitation, it is removing dropped fruit, and also pruning of the plants that are infected, where we have like the branches that are presenting symptoms. Pruning these branches can reduce the inoculum source and kind of controlling a little the disease. So moving forward to advance the management of leprosis and what our group here is doing, we are trying to identify the viruses and the vectors. With these, we can design target control strategies to know what we are dealing with and control the correct species of vectors and avoid them to spread the virus to other crops. And another thing that we are testing and have some research on is on biological control, that is trying to use predatory mites in combination of acaricides to control the Brevipalpus urticae. We do have some studies with this for hibiscus plants. And we found some species that has potential to be used as an adjuvant with the acaricides that are not going to be highly impacted. So then together can be used in IPM and have a better control outcome. I'd like to finish by thanking many of our collaborations and by the institutions they are partnered with and our team here in South Florida. And that's it for this presentation. So if you have any questions, please type on the chat or we can discuss this in the end of the section.
Thank you, Eileen, for a wonderful presentation. Chris and I and Tara and everybody really appreciate you for stepping up and delivering Dr. Carrillo's talk on his behalf. So thank you so much. We'll wait to listen from you again for another talk. For the next speaker, I am going to introduce Tara Pittman. She's also at UC Davis Contained Research Facility. She's working with Dr. Chris Godfrey, and she's going to talk to us, give us update on citrus leprosis research activities at the UC Davis Contained Research Facility. Tara, you can start sharing your slides and take it from there.
All right. Thank you for that introduction. I'm just going to do one other thing. There we go. All right. So I have been working with the mites and viruses under laboratory conditions to determine transmission. Specifically, I work at the UC Davis Contained Research Facility, and it's a facility that allows for us to work with quarantine pests and pathogens without having the possibility of introducing those in California agricultural systems. So it is a BSL level 3 specific to plant pathogens. And although most of the mites that we're working with are native to California, it's the viruses that we want to keep in a contained environment. So the research overview for what we were doing with this pathosystem. We really wanted to know if Brevipalpus mites in California are capable of transmitting or vectoring citrus leprosis viruses. So we focused on the 2 species that are most commonly found in citrus throughout most of the state, which are B. californicus and B. lewisi. And then the leprosis viruses that we were primarily interested in were the ones that we felt might have a higher risk of coming into California through various mechanisms. So those were citrus leprosis virus cytoplasmic type, CILV-C2 also, and orchid fleck virus. And as the previous talks discussed, we were mostly concerned about those because CILV-C is currently found in Mexico and could come up with citrus shipments. And then CILV-C2 and orchid fleck, because those are present in nursery industries for ornamentals and have, in the case of orchid fleck, have occasionally been found in orchids in California nurseries. To be able to do this research, we also had to be able to compare them to known vectors. So we received Brevipalpus yothersi from our colleagues in Florida as the known vector for both CILV-C as well as CILV-C2. So once we received the 3 different mite species, we had to figure out how to cultivate them under laboratory conditions. Maybe you wouldn't think about it, but these things can actually be fairly difficult to keep alive in a controlled environment because you have to worry about things like humidity, temperature, as well as having palatable food supply. So, and also one of our big hurdles was the fruit we were receiving sometimes had predatory mite eggs on them. And under laboratory conditions, those predatory mites had a really easy time and could actually decimate all of the mites on a fruit, which is possibly good news for growers, but sometimes it really set back our research goals. So we had to maintain the mites on the fruit because it is the easiest way to transfer mites from one surface to another. It's also the easiest way for transferring virus material. So it was easier when we were able to get fruit that had lesions on it to do this work than it was with other material types like leaves. As backups, we maintained mites on little citrus seedlings. And that way we didn't have to worry about transferring them as often because the seedlings would live for a much longer period and would finally decline just due to overpopulation with mites. But the difference is on an orange fruit, a mite colony can keep going for 2 or 3 months, whereas on a citrus seedling, we have some that have been going for 9+ months. So it's just a way to keep mites going without having to think about them quite as much. So here's an example of a fruit colony. So on the left, that is the full navel orange, and all of the lighter orange is the fruit itself, and that darker reddish orange is actually accumulations of mites, both mobile stages as well as eggs. And the upper right photo is a group of adult mites under magnification, and the lower right image is eggs under magnification. So we can get pretty high population densities in the laboratory. And here's an example of those backup colonies on a little citrus seedling. The upper image shows adult mites walking around on the lower leaf surface, and the bottom image to the right shows the stem with that prolific feeding damage and adult mites and eggs. So things that we had to consider to conduct this research included the fact that mites are very small and they are very soft-bodied. So we would have to transfer a lot of mites to make sure that we kept most of them alive. So we made sure we transferred at least 100 individuals per replicate per experiment. And then we would also periodically check for mortality in the experimental groups. So once we transferred them, we would go back and look at the fruit or whatever half a day later, and then the next day when we would go back to transfer again, then we would see how many were still alive. And generally, mites take at least 4 hours to acquire citrus leprosis viruses from feeding on plant material. But they don't necessarily settle down to start feeding as soon as you transfer them onto a surface. So we generally allowed 24 to 48 hours for an acquisition access period to make sure that they had ample time to settle down into feeding behavior and ingest long enough to be able to pick up the virus. We also had to do molecular detection to see if the mites have the virus. So throughout the talk, we've been looking at symptoms on plant material and fruit, but there's no way to visually determine whether a mite has the virus. So to do that, we had to actually destroy mites and do molecular testing because there's no visual discrimination. The first virus that we did experiments with was CILVC. We had a collaborator down in Tabasco, Mexico, who was able to go into the field to collect fruit and then ship it to us at the CRF. So what we did was we received the fruit, we would open in a biosafety cabinet, we would inspect the fruit for other mites, as well as any other potential insects, and clean the fruit with soap and water and isopropanol to make sure any incidental organisms were off the fruit. And then we would move our research mites onto individual pieces of fruit. So for CILVC, we determined that 24 hours was an adequate period of time for virus acquisition. At that 24-hour mark, we took samples of mites to complete RT-PCR with to determine if the mites were in fact feeding on the lesions and picking up the virus. And then to determine whether the mites were retaining the virus, as in they were becoming infected themselves with the virus, we would take another subsample of mites 4 days after moving them onto a clean piece of fruit to see if those mites still had the virus. And then to determine whether they were able to transmit the virus to citrus hosts, we also took mites from that lesion that had fed on that lesion material and put them on citrus seedlings and left them for 1 month. So here's an overview of what those experiments looked like. So we would start with adults because the adults are the largest version and they are mobile. And so they were the easiest to transfer between fruit and onto plants. So day 1, we would take adult mites from colony fruit. We would move them individually over to lesions on infected fruit. 24 hours after that, we would test for the virus with a few of them. More of them would be moved to clean fruit, and more of them would be moved to healthy seedlings. From the clean fruit, 4 days later, we would test those for the presence of the virus. And then for the ones that were moved to the citrus seedlings, those were left for 30 days to allow them to settle in, feed in lots of places, and then we would check them for the virus, as well as visually examine those seedlings, look for feeding damage on leaves, and look for lesion formation on leaves and twigs. All right, so at the very end of the experiments with the plant material, what we were looking for was feeding damage on leaves and any lesion formation. So on the left, this is an example of the signs of the presence of mites and feeding damage. So there are some little flecks across that leaf surface as well as cast skins. So I would know that on this leaf, we had mites that were feeding, that were present for long enough that if they had the virus, they would have transmitted it. And then on the right is an example of CILVC that was transmitted by mites onto this orange seedling. So it was very typical ring-like spots with that little bright yellow foci, and then ringed in some lighter green. So the results of the experiments for virus retention, meaning whether the virus was able to be picked up by mites that were feeding. Here we're seeing that the clean colony mites clearly do not have the virus present before being introduced to viraliferous fruit. After feeding for 2 days on the lesion, all 3 species tested positive for the virus, which means that all of them were feeding on that fruit surface and they were feeding on lesioned material. So they were interacting with the virus in that plant host. Once those were transferred to clean citrus fruit, 4 days after feeding on that clean fruit, the only virus species that was still testing positive for the only mite species that was still testing positive for the virus was B. yothersi. Both B. lewisi and B. californicus did not retain that virus after being removed from that virus source. So we proved that the known vector does in fact retain the virus, whereas our 2 native species do not retain the virus. And then the ability to inoculate the virus to citrus seedlings. So this data represents both the virus, the mite species, and the citrus seedling after 30 days of feeding by potentially viraliferous mites. So you can see that B. yothersi mites and the seedlings they fed on were positive for the virus at 30 days, whereas B. lewisi and B. californicus, the mites did not maintain the virus and they did not transmit it to that citrus host. So in review, B. californicus and B. lewisi were able to ingest the virus while feeding on lesion fruit, but they did not retain it and they did not transmit it. B. yothersi, on the other hand, both picked up the virus and transmitted it to the citrus seedlings. And taken all together, we feel that this strongly supports the fact that B. californicus and B. lewisi are not vectors of CILVC. The virus that we're working with next is CILVC2. Our Florida collaborators, again, sent this to us, and we received viraliferous lesions on hibiscus leaves. The big issue that we ran into starting with this material was that our California species don't like this as a food source. We would move mites onto it and they would just crawl off and die. So we were only able to initially work with B. yothersi to be able to get this into citrus leaves on those little citrus seedlings. And so we are now at the stage where we have viraliferous B. yothersi on citrus seedlings. And those citrus seedlings have leaves and stems that do have lesions on them. And so we're going to proceed with trying to do the same types of experiments with this virus that we did with CILVC. And the third virus that we have managed to work with a bit was orchid fleck virus. So we were able to receive some orchid plants from CDFA. They were positive for orchid fleck virus. The image on the right is a leaf being held up to the light, so you can see those lesions with a light halo around them indicating symptomatic lesions. There were brevipalpus mites present on those orchids, and they did test positive for orchid fleck virus. We took those orchid fleck virus infected mites and moved them to orange seedlings. But we were not able to find lesions on any of those host plants that they were moved to. Those seedlings never developed any signs of lesions. None of the material that was showing signs of feeding damage tested positive for the virus. And so we are fairly certain we did not do the appropriate sequencing to determine which strain of orchid fleck virus this was, but based on these experimental results, that seemed to be a strain of orchid fleck virus that does not infect citrus. So we are going to continue to look for citrus infecting strains of orchid fleck virus. And if we ever find that, then we'll proceed with more experiments. So in summary, CILVC is a risk for continued northern spread, possibly to California, but the primary mite species through California cannot transmit the virus. So it is unlikely in those orchard systems that this would be able to be established. We still have experiments pending for CILVC2, and hopefully we will at some point have good experimental results so that we can make a statement about that one way or the other. And then orchid fleck virus has been found in California orchids, but it is a quarantine pathogen that CDFA goes in and destroys plants where it's found. And also the fact that not all orchid fleck virus strains go to citrus suggests that this is hopefully a low risk mode of movement into California. But CL viruses can infect multiple plant species. For example, we received material on oranges, hibiscus leaves, and orchids. So everybody needs to stay vigilant as to the possibility of this moving into California on ornamental species. And that is my talk. Thank you.
Thank you, Tara, for that excellent presentation. With that, let's move on to our last topic for today. Ellen Tassi from University of Florida is going to speak again, and she's going to speak on a topic updating us on taxonomy of the mites, brevipalpus mites. So, Ellen, back to you again.
So now I'm going to talk a little about the updates in the taxonomy of flap mites. Just an overview, when we talk about mites, they are in the phylum Arthropoda with the insects. Then they divided, the insects go to the Hexapoda and the mites go to the Chelicerata. It's two different branches. The insects, we have around 900,000 species described, and mites, we have 55,000 species described. There is the estimative of whether we have more or less 1 million species to describe in mites, while we have 2 million to describe in insects. So there is a really unknown diversity in this group. When we think about mites, there are two main branches in their taxonomy. One branch is the parasitiformes, where we have the ticks, for example, and all the species that feeds on mites that feed on plants, that are the phytozoite, for example. And in the other side of the branch, we have the acariformes that contains the mites that are going to feed on plants. Within the acariformes, we have the prostigmata, and in the prostigmata is where our family of interstitium palpide is located. Temipalpidae, the whole family today has more than 1,000 described species. Like generally, when we talk about Temipalpidae, their basic morphology, they are small mites, 0.2 to 0.5 millimeters in size. There is a wide variety of colors. They can be yellow, green, orange, translucent. The Brevipalpus ones that we worked with, they're more orange to reddish. All of them have a long life cycle with 60, 90, even more days. All of them like to lay eggs in crevices associated with trichomes, fruits, stems, or lesions. So they kind of protect their eggs. All of them are rounded, shaped, oval, and have at least one or both sides of the body flattened. That's why they have this name, flat mites. Between the genera, the most relevant ones, we have these genera are the most relevant ones, the ones that are most reported causing damage to economic important crops. We have the Hauiella, the Temipalpus, Cenopalpus, Brevipalpus, and the Dolichotetranicus. But between those, the most important is the Hauiella. There's the red palm mite, that's a direct pest because it causes damage by feeding and is an invasive species. And the Brevipalpus, that are the ones that vector plant viruses. Again, when we think about Brevipalpus, they are in reality the second most numbered genera in Temipalpid right now because they have a lot of new description of Temipalpus. But even so, it's almost 300 species. They are mainly distributed in the tropical and subtropical areas. But there are described species from the Nearctic and the Palearctic regions. They have this pest status because of they feed on crops and ornamental plants. So the Brevipalpus also can cause damage by direct feeding. For example, this is soursop, where we have high populations in Brazil. These pictures were taken in Brazil. With high populations of Brevipalpus, you're side feeding that can cause disease scarring on the fruits. This kind of damage is also reported in lemons and cacao plants, for example. So when we have like high populations, it also can cause damage by direct feeding. But the main problem is when they are vectoring viruses, as all the viruses that we talked about in the previous presentations. Another problem with the Brevipalpus is because they are regulated quarantine pests. For example, Brevipalpus chilensis, lewisi, and californicus, they have restrictions by the international market, especially for fresh fruit. This can cause a lot of losses. We have to control. We can lose shipments. We can have all the shipments have to be treated before entering a place because of the presence of these mites alone. So the first step from any study is understanding and correctly identifying the pest that we are working with. So is the main thing. The first thing that we have to do is correctly identify the mite before doing any transmission assays or anything, because we need to know what we are dealing with. And this data will support all the development control strategies, because we have to work with the correct mite to do all the experiments for control strategies, and also to adopt the corrected quarantine measurements. We have to have barriers in place for the species that are really the ones that are going to cause damage or transmitted virus, not the other ones that are innocent guys. But the taxonomy of this group is a challenging puzzle. Why? Because they are really small. There are not many characters that are look-alike. So there was lack of study in their morphology. All the studies are based on few characteristic things. And so there's a lot of confusion during the years. So right now we are literally reviewing most of the description in the world reports. And we also now have the molecular phylogenetics advancing and helping us to guide these new studies on systematic of this group, and this is what we are doing. Currently, what we have is that there was a a system when they divided the Brevipalpus species by counting the number of city and other characters in 6 species group. This division was proposed in 1987. But these were, like I said, like you counted the number of structures in the legs, the number of city, the format of the city, and that was it. But what the problem of this organization, is that it's artificial. It would not allow us to have any prediction on the bioecology and the relationship evolutionary aspect of this group. So right now, what we are doing, we are planning and starting to do new kinds of studies that are gonna create the possibility of us to create better provision on this group. So one of the first steps is do a detailed morphological study. Now we have new different kinds of microscopy than the ones that they have in the past. For example, we are using differential interference contrast microscopy and scanning electron microscope. These allow us to see more details in the mites, so we have more characteristics to work with. With these, we could see new morphological traits that we are using now, like the shape of this vesicle that is called spermatheca, is where the sperm of the mite is deposited. It is there, unlike, even if they are not reproducing sexually, is a structure that they kept. These mites also have in their cuticle, in their surface, some structures like these. They're rounded structures that are called microplates. And by studies using scanning electron microscopy, what we could see is that different species have different patterns of reticulations on these microplates. And we are also incorporating catataxia and ontogeny on these studies. All of this together led to, in 2015, we have the first review in this genus, that is the Brevipalpus phoenicis. When they studied these species, they realized that this was in reality 8 species that were grouped under the same name, including species wrongly killed by science that were resurrected, and new species that were cryptic inside this name. So they reorganized it, and with this we could restudy it, and that's why now we know that Brevipalpus, the other side inside the Brevipalpus phoenicis complex, is one of the main vectors. So our next step now is using other species complex, study other complexes, and doing an integrative study using deposit material, new material, and molecular studies to try to put light on this taxonomy. So moving on, our strategies for this work is increase the sampling, doing morphological studies with the new material that we are sampling and comparing with the holotypes that are deposited, the majority of them in the Smithsonian collection, and doing DNA sequence of multiple molecular markers to build phylogenies, and then doing an integrative analysis of morphological traits and molecular data. We do the molecular data, take the data and compare with the morphological to see if we can make sense of it, and use the data from the morphological. One supports the other, so it's a both way patch that we keep checking one with the other to see if what we are thinking is the correct distribution and grouping of the mites in a species is correct or not. In the morphological studies, for example, as you can see here, we are using all those techniques as light microscopy, scanning electron microscopy, comparing with the type deposit. And as you can see here, those are all different species. They differ on the reticulation pattern, for example. So we are looking like every little detail on these mites to try to put light and understand what they are. What we are doing right now in our lab is finalizing the review of the Brevipalpus californicus group. When we think in a species group, the ones that have the counting number of characters, that old group distribution that was created by Baker in 1987, we had 23 species that were Californicus species group. He studied all this type material, realized that in reality it was 46, so there is already a mistake here. And then we focus only on Californicus, the real Brevipalpus californicus, not the group. So everything that was deposited on this collection under Brevipalpus californicus investigation, we went there and restudied all these types. And we have something around 8 to 9 species that we are working on. Some of them were cryptic and we're describing because they're new to science. Some were wrongly synonymized, so this is what we're working with. So yeah, this is what we have, around 9 species under this Brevipalpus californicus name. And after having this, we can see which are the ones that are associated to the plants that have the symptoms, at least here in Florida, this will be our strategy. And then we can try to go back to transmission assays like Tara showed you, and then we can see like which are the mites, at least here, that are the ones that are responsible for vectoring the viruses. So this is one example of what we are having on phylogenetic analysis. Our data set was not that big. We included just 15 species of the 300 were the ones that are most important and commonly found on the plants that had virus associated. So what we can see that the molecular markers, then the combined of 4 markers, we can see like really good grouping. So it's all the branch here are monophyletic, so it makes sense that what we are doing is a good work. The separation is going nice, but this is what we are finding. The Baker species groups, for example, the Phoenices, we have species here that they are not corresponding exactly to what we are finding when we think about phylogenetic groups. For example, the original Phoenices group has Yodersae, Phoenices, Papayensis, and in reality, they separate here. They show up in here and here, for example. So they are present in more than one branch in the phylogenetic tree when we're using molecular markers. When we do other characters like the microplates, what we can see is that they are a really good morphological trait to see these groupings. They are more related, like species specific and not group specific, but it's a character that they are using to support it. So probably are going to incorporate it to create the groups, new groups, not only the characters that Baker was using, like counting structures. We're going to use other structures. Another important feature that for us is the most informative, that links the morphology with the molecular data, is the shape of the spermatical apparatus in the females. So different groups, like this Yodersae group here, all the spermatica have the same general shape. So all the species that are inside this group has this shape of spermatica. The same was observed for the Californicus group and other groups. So this shows up as a really important character for us. And here show like the original phylogenetic, the original morphological trait in the species groups, like counting the number of structures. We have like the Brevipalpus Phoenices as being a polyphyletic because it ramifies in 2 parts of the branch. This can change when we add more species because, as I told you, we have just 15 in this analysis, and in reality we have 300, so we don't know how much spread we're going to have here. But all of these are be doing right because we want to use these to make inferences of the other data, like what branches has vectors and what branches has no vectors. For example, this branch here, it's going to group Yodersae, that is a vector, Papayensis californicus, that they are vector species. But in this middle, in the same branch, we have Chilensis coming out. So with this, we can predict that there is a possibility that Chilensis could also be vector of Brevipalpus transmitted viruses. As in reality, recently, it's not published yet, but yeah, we have found some viruses in Chile associated to this mite. So this is one thing that help us to make predictions and go to the reality, like the field, and see what we're looking and try to better understand in the system. The other side, like these species in the bottom of the phylogeny, like the what we call Cuneatus group, these species There's no reports of virus associated to any of them. And as we can see, it's coming out in a separate branch. So could be this an indicator. Another thing that this kind of studies tells us is how to look like probably groups of polyphagous, oligophagous, and host-specific species. Like here on the other side, this branch, we have all these species being polyphagous species, and it's confirming real ecological data. This other group that has papayensis, phanesis, obovatus, we have species that are known to be oligophagous, and they are all grouping. And here in the bottom, the cuniatus, we have species that we know that are host-specific. So this is guiding us to do the ecological, morphological study, putting it all together. Another thing that this kind of study is showing us, as you can see here with the Brevipalpus ursi 1, 2, Incognitus 2, is that we have more cryptic species than we previously imagined. Some of these species we look at, they have some morphological traits different, but we're not sure if they are, this is just a variation inside a species or if it's really a new species. One problem is since they are parthenogenetic, we cannot try to mate them to see if they produce viable offspring. So this is one tool, another thing that makes it more difficult to study these mites. And using this integrative approach, we are seeing that some small variations that we are seeing morphologically really represent new species when studying them using molecular data. So in general, of my considerations for this presentation is that we are doing the first step in the phylogeny of Brevipalpus. We are pointing out new important morphological traits to identify them. With this, we are revealing a lot of cryptic species and diversity new species are emerging. With all this data, we are being able to create hypotheses in evolutionary aspects to try to look for viruses and other things. So the morphological data and molecular data support each other. So all of these are going to probably in the future support identification, or we can have like really specific markers that we can develop in the future to like immunostrip assays to identify the species that we have. The species of mite that we have is the one that vector the virus. With these, we can do better predictions of the virus spread and also improve quarantine and biosecurity measures, like targeting specific the important species, the ones that are going to vector viruses. All of this is essential for integrated pest management because all the other more applied studies depend on starting with the correct individuals that have to be the control. Also, we can target the crops that are in higher risk of being affected by these mites. So we can make vigilant, like for crops that we know that has Brevipalpus and there are viruses that infected them. For example, hibiscus and ligustrum that I told you, there are several mites that like this plant, Brevipalpus mites, and also virus circulating on them. With these, we can improve the surveillance and also support international trade by reducing disputes over what is a pest and what is not a pest. So I'd like to thank you, the group again. It's the same slide as before.
Thank you, Ellen, for that presentation. With that, we have finished all our presentations for today. All the speakers have given their presentation. And like I said before, there were some questions in the Q&A session which we have answered. There was one question and Chris marked it as Chris would like to answer the question live. Chris, would you like to read the question and answer it, please?
Yeah, let me pull the question up just a sec. Okay, this is from Heather Martin. And it's one interesting idea is that leprosis is a mite virus and citrus is acting as a vector. Can you get virus from Hawaii where they have found orchid fleck virus in citrus? Also, orchid fleck virus has a B rating. It's regulated in nurseries, but not a quarantine pest. So in response to that is I have all the necessary permits to get these viruses from either Hawaii or Florida, and I can get them in plant material, and I have to make a modification to a permit for Hawaii to get a Brevipalpus mite that may have it, okay? But some of the problem we're having has to do with the number of strains of orchid fleck virus that are out there. And so what we've been hunting for is trying to find a virus we know goes into citrus and has a brevipelpus species we can get with it. So we're trying to answer this question, Heather. It's just we're not quite there yet. Sorry for that answer, but that's kind of where we're at.
Okay, there were a couple other questions that I thought might be helpful to answer, like talk about live. One of the questions, and this Ellen answered that, so I'm going to call on Ellen. Ligustrum, a common landscape plant, should we be monitoring for that for virus or mite? Ellen, would you want to, because you are the one who answered this question?
Ligustrum plants, we have several species of brevipalpus that likes ligustrum, that feeds on ligustrum. And we also have several viruses that I showed in the first presentation that goes to ligustrum, and ligustrum can also be affected by passion fruit green spot virus and citrus leprosis virus in natural landscape, like we found this in Brazil, for example. So I think this is a really good plant to be taking a look at, as the same for hibiscus and orchids, as we said before. I would try to keep a look for especially the presence of the symptomatology. That could be a really good indicator of the presence of this virus in the landscape.
Thank you, Ellen. Devin Carroll asks, would you recommend sulfur for controlling mites? Well, it is one of the recommended products for controlling all kinds of different mites, but it is also a broad spectrum that controls natural enemies. So use it wisely. There are other safer products that are more softer on natural enemies and would control mites. So to answer Devin's question, yes, you can use sulfur, but there are also other safer choices. There were two other questions that I think I want to bring up. One of the questions is how long the disease can last on a tree. Is it still active in the following year? And Tara answered this question. So Tara, do you want to take it, or I can read your answer?
Go ahead and just read my answer.
Okay, so Tara said the virus is active as long as the infective cells are alive. And so that means that it can be there in the next or the following season too. Lesions on twigs and branches are more likely to last longer, including into a new growing season. Lesions in leaves and fruit tend to drop within a few weeks. So, if there is lesions on the branches and twigs, that can serve as your inoculum for the next season, which brings me to another question: why pruning helps? There was another question: how does pruning help? That is exactly how it helps, by removing the inoculum so that you don't have, but the mites would have to feed in order to transmit the virus. So if you reduce inoculum by pruning the trees, pruning the tree, cutting out the infected branches and twigs, that helps. I think those are the other two questions that I wanted to discuss. Okay. Tara, there is a question in chat for you. Not in the chat, in the Q&A section. Do you want to take it?
In the Q&A. Sorry, I didn't see that one. Yeah. So the mites tend to take on the colors of what they are feeding on. So when they're on fruit, they tend to be bright orange. When they are on plant material, they tend to have kind of blackish or dark green spotting in them. So it really, it just depends on their food source, kind of what color they end up being.
Okay. I just want to thank everybody. Peter, Cheryl, thanks for organizing. All the speakers, thanks for accepting the invitation to speak. And all the attendees, thanks for attending. We're going to close it now then.
Yes, yes. All right. Thank you, everybody, and have a great rest of your day.